amino acid, which is urocyanuric acid and carnic acid, which control and maintain the acidic pH of the skin, which is very important to maintain skin hydration and integrity of the skin barrier, and control trans-epidermal walls. And this eventually maintain hydration of the stratum corneum, which is very important, as we will see, inshallah, in the next slide. And this represent the natural moisturizing factors of the skin. Moreover, filigree causes organization of the epidermis into compact structure. And this is going through the layer of the skin, the epidermis and the dermis. What's concerning us so much in the atopic dermatitis is the granular layer with the presence of the tight junction, the stratum corneum with the content of the intracellular lipid, the keratinocytes, skin microbiota. So let's see every one its job exactly. The tight junction regulates the trans-epidermal water loss, transports of iron and solutes. Keratinocytes produce antimicrobial peptides, the key role in pathogen clearance and maintenance of tight junction integrity. And this stratum corneum intracellular lipids represent the mortar is زي الأسمنت. In the brick and mortar model, the stratum corneum with the intracellular lamellar bodies represent like brick and mortar model, like double asmant, to maintain its integrity and prevent its penetration and dysfunction. To maintain stratum corneum hydration, integrity of the skin barrier, preventing the most dangerous organism, in addition to others, colonization by staph aureus. What about the skin microbiota? It contributes to the immune homeostasis through preventing colonization of the staph aureus and maintain normal immune responses, preventing skin inflammation. So let's see what's going on in atopic dermatitis. بيحصل skin barrier deficiency through a lack of filigree, abnormalities in tight junctions, altered composition of the stratum corneum intracellular lipids, and deficiency of the antimicrobial peptides. Lack of filigree is known to be decreased in the epidermis of the atopic dermatitis patient, and approximately 25 to 50% of the atopic dermatitis patients carry filigree mutations, especially in patients, and this is very important, who have early onset atopic dermatitis and those with persistent severe cases. However, about 40% of individuals with filigree mutation, even null alleles, null mutation, do not have atopic dermatitis. Therefore, we can get an filigree mutation alone does not stand for the occurrence of atopic dermatitis. It is interaction. However, it is the strongest risk genetic factor for the development of atopic dermatitis. Filigree deficiency, it is very important to know that it could be genetically determined, whether it is haploid insufficiency or non-mutation, or it could be secondary to the skin altered immune response causing filigree deficiency. But filigree deficiency ممكن تبقى genetically determined non-mutation أو مثلا secondary to the heightened immune response. Now, what are the consequences of filigree deficiency? It allows allergens and pathogens to penetrate the epidermis and induce inflammasome and proteases activation, with subsequent heightened altered immune response, promotes microbial colonization, and causes tight junction disruption to decrease expression of the scaffolding protein, making this tight junction in the granular layer. So what about the altered composition, the morator, in the Brica morator model of the stratum corneum? It causes the decreased intracellular causing dryness of the skin and increased trans-epidermal water loss. So what about the deficiency of the antimicrobial peptide? This keratinocyte, the defective immune response and the continuous chronic inflammation, causing the keratinocyte to decrease its production of antimicrobial peptide, and it is linked to high susceptibility of staph aureus colonization other skin dysbiosis like malassezia, like viral infection, and exacerbation of atopic dermatitis by accentuating the immune inflammation. So it ends again in accentuating the altered immune response. So we can see in this figure that the disruption of the skin barrier causes penetration of the allergens, organelles, and other viruses, and going down into the epidermis, stimulating the Langerhans cell, which act as antigen-presenting cells, and stimulate the keratinocyte to produce pro-inflammatory cytokines that will end into stimulation of the T helper 2 and ending in stimulation of B cell, reducing immunoglobulin E, as well as the multiple T helper 2 pro-inflammatory cytokines. So skin barrier and immunological derangement, and immunological derangement will cause more and more skin barrier disruption. Both innate and adaptive altered immune responses are involved in the pathogenesis of atopic dermatitis. However, T helper cell is the key pathogenic factor in the immune dysregulation observed in different stages of atopic dermatitis, and mainly it is helper 2 cells throughout the entire course of the atopic dermatitis. However, other types vary according to the stage of atopic dermatitis, as we will see inshallah in this figure. So going through this figure, we see that this is a non-lesional skin in a patient with atopic dermatitis with somehow of mild disruption of the skin barrier, activation of the T helper 2, together with a state of microinflammation and increased number of the T helper 2 cells. However, in the lesions of acute atopic dermatitis, there is penetration of the allergens and pathogens, stimulating the Langerhans cell, making it functioning as an antigen-presenting cell, and stimulate the production of thymic stromal lymphopoietin, interleukin 25, interleukin 33 from the keratinocyte. These cytokines will act on the naive CD4 T cells, causing them further differentiation into T helper 2, either directly or through stimulation of the innate lymphoid cells too, causing activation of the T helper 2. The T helper 2 will produce its pro-inflammatory cytokines, interleukin 4, interleukin 5, interleukin 13, which will act through the JAK-STAT signal, which it will signal its effect through the JAK-STAT pathway. Moreover, the T helper 2, the activated lymphocyte, will produce the phosphodiesterase. This phosphodiesterase is a part of the phosphorylase. It converts the cyclic AMP, adenosine monophosphate, into adenosine monophosphate. But what about the phosphodiesterase? It produces paramount secretion of these pro-inflammatory cytokines. And we can see this finger, here, scratching. This scratching will cause more and more disruption of the skin barrier. And the T helper 2, its production of interleukin 31, will induce a vicious circle of more and more scratching, causing more and more disruption of the skin barrier and entry of allergens and pathogens. This T helper 2 will act on the B, stimulate give help to the B cell to produce immunoglobulin E. So this is in the acute stage. The predominant immune cell is the T helper 2, acting directly or through giving help to the B cells to produce immunoglobulin E, resulting in sustained inflammation. In addition to the activation of the innate immune system and production of cytokines that exacerbate scratching, making the stages a vicious cycle. Going to the late lesion in the chronic, again, there is abundance of T helper 2 cells, but because of the excessive microbial colonization and the disruption of the skin barrier, it comes to produce T helper 1 cytokines, interferon gamma, tumor necrosis factor alpha, fast ligand, which induce the differentiation of the naive T cell into T helper 1 cell. Furthermore, there is production and differentiation into T helper 17 and T helper 22, which produce interleukin 22 and T helper 17, which produce interleukin 17. However, the role of T helper 17 in atopic dermatitis is still under investigation, and some going to the third mechanism in pathogenesis of atopic dermatitis is skin dysbiosis and colonization with the Staph aureus. This affects the skin immune response and induce inflammation. Staphylococcus aureus can also secrete a number of proteases, enzymes, and cytolytic toxins that induce cell injuries, engage pattern recognition receptors, and cause skin inflammation. This is a very important mechanism involved in the pathogenesis of atopic dermatitis. Another mechanism is the itch scratch itch cycle, which is part of the atopic dermatitis clinical presentation. However, it is just not a symptom. It's very important in the pathogenesis, the cycle of pathogenesis of atopic dermatitis, because it further promotes inflammation through release of mediator. This mediator stimulates the nerve fiber and regulates cellular protoregulants through several H mediator, and their corresponding receptors are reported to contribute to itch, including mainly histamine, and it acts through its receptor, histamine type 1 and type 4 receptor. Substance B and neurokinin 1 receptor, these are its receptors are expressed on eosinophils, T cells, and the peripheral nerve fiber, causing severe pleuritis. Interleukin 13 just shortly, which is the T helper 2 cytokines, act through peripheral nerves. And interleukin 4 and interleukin 13 act on the sensory nerve and stromal lymphopoiesis. So it is a skin barrier deficiency, scratch itch cycle, skin dysbiosis, and again skin barrier deficiency. The genetic and epigenetics, why it has a strong genetic predisposition, because the concordance rate of 72 to 86% was observed in identical twins, and up to 23% in dizygotic twins. The main genes involved is the fibrine and the serine protease inhibitor Kazan type 5, which is well involved in the Zartor syndrome, in addition to other genetics and methylation, hypermethylation process for the genes controlling the cytokines production and receptors. Environmental factors include breastfeeding and the time of weaning, because delayed weaning was found to be associated, which is somehow strange, but it is somehow depend on observational studies, not like the solid science. Obesity, pollution, exposure to tobacco smoke, which is expected because it will disrupt the skin barrier, excessive choline and calcium carbonate in water, antibiotic usage because it will end in skin dysbiosis. Daycare attendance and early exposure to vets may help against the development of atopic dermatitis. ودي كانت محاضره جميله في المؤتمر اللي فات، الدكتوره بشرى قالتها انه ممكن الاكسبوجر للفاتس ممكن يكون عامل بروتكتيف اجينست الديفلوبمنت اوف الرجيك ديسوردر، عشان الهايجين هايبوثيسيس. ان كل ما انت قفلت على الناس بيبقى الموضوع ايه، ممكن يبقى نيور اكسبوجر للالرجين او الباثوجين يخليها تعمل سكيود ريسبونس تورد التي هيلبر 2، لكن الايرلي اكسبوجر للمايكروبز هيخليها تعمل ريسبونس يوزنج مين؟ تي هيلبر 1. So in summary, this is exposome. It means that exposure to environmental factors together with genetic and epigenetic regulation. Preclinical, it's some sort of subclinical skin barrier dysfunction. It then passes into stimulation of the innate immune response, and then passes into a stimulation of the adaptive immune response with predominance of T helper 2, and then later phases T helper 17 and T helper 22, exacerbated by each scratch cycle and the skin dysbiosis, and together with, in some patients, substantial percentage immunoglobulin desensitization, ending in the clinical presentation of atopic dermatitis, the non-atopic comorbidities, and in some patients, immunoglobulin E is presented to the self-skin antigen, ending in autoimmune variant of atopic dermatitis. We'll start بكره ان شاء الله فالدنيا يعني كويسه باذن الله. Starting which of the following is a false statement about the immune alteration underlying atopic dermatitis? T helper 2 cells predominate in the entire course of the disease. Interleukin 4 and interleukin 13 are pivotal in its pathogenesis. Immunoglobulin E mediated hypersensitivity is the main underlying mechanism. Skin barrier deficiency exacerbates the altered immune response. Thymic stromal lymphopoietin activate T helper 2 both directly and indirectly.
يا ريت اللي بنوجه الحساسيه والمناعه ما يجاوبوش لان هم طبعا عارفين الاجابه. هيسيبوا الناس بالتصنيف.
3. Which of the following is true about filagreen in atopic dermatitis? Filagreen deficiency is always genetically determined. It is responsible for the alkaline pH of the skin. It is produced by the innate lymphoid cells. Filagreen deficiency triggers skin inflammation and dysbiosis. Non-mutation in filagreen is correlated with adult onset atopic dermatitis. Which is the following is true. True. احنا
لا لا بعد ما يجاوبوا
طيب
ما تقلقيش.
هي اول مره هيكون بالاول واحده. Four. Yes. Because when we said that the filagreen deficiency can be secondary to immune alteration, it is responsible for the acidic pH, which is essential for maintaining the skin barrier integrity. It is produced by the keratinocytes, and non-mutation in filagreen is correlated with early onset atopic dermatitis and in severe cases. About the itch cycle in atopic dermatitis. Thymic stromal lymphopoietin is one of the triggers. Histamine induces itch, particularly through histamine receptor 2. Interleukin 4 and 13 can directly activate sensory neurons. Substance B and neurokinin 1 receptor are the main pruritigenic. Interleukin 31 signaling triggers itch via JAK state activation. Number 2, it actives through histamine actives through 1 and 4. At the end of my lecture, I would like to thank والله بشكرهم جدا كل اساتذتي وزملائي في الوحده وربنا يرحم الدكتور يحيى ويجعل كل ده في ميزان حسناته لان هو فعلا provide for us مكان يعني مش متصورين التعب اللي حصل فيه قد ايه هو اساتذتنا عشان يوصل للحاله اللي احنا فيها الحمد لله رب العالمين وربنا يديم ويقدرنا ان احنا فعلا يعني نحفظ الامانه باذن الله. And thank you وفكرت
Thank you very much Professor Dalia for this very informative talk. الحقيقة والتزمت بالوقت.